This video is intended for BSc Biotechnology 4th semester students, and the subject is genetic engineering. So in some plants, it is observed that a cancerous outgrowth is formed if one of its stems suffer from some kind of injury. This is due to a bacterium known as Agrobacterium tumefaciens. It is referred to as nature's smallest genetic engineer because it inserts a DNA molecule inside the plant body known as tDNA. So this tDNA resides in a plasmid known as TI plasmid or tumor-inducing plasmid. tDNA contains genes that code for enzymes synthesizing opines and phytohormones like auxins and cytokinin. Opines are low molecular weight compounds that provide nutrition to the bacterium. Apart from tDNA, the plasmid contains other regions such as virulence regions, which help in tDNA transfer into the plant. Next type of gene is opine catabolism gene that produces enzymes necessary for the utilization of opines by Agrobacterium. And there is original replication, which is required for the propagation of Agrobacterium. tDNA is a 23 kilobase segment which is transferred to the plant genome during Agrobacterium infection. As you can see in the figure, the tDNA contains two regions, left border and right border region. As I already told you, tDNA is involved in the synthesis of plant growth hormones that lead to the overproduction of those hormones in plant body and ultimately cancerous growth. Apart from tDNA, it also contains some eukaryotic regulatory sequences that helps in expression of bacterial gene in the eukaryote, that means plant. Coming to virulence region, it contains 8 operands and altogether 25 genes. The function of vir region is to transfer tDNA into the plant body. So there are many steps which are involved in infection, and the steps are cell to cell recognition and binding, signal transduction and transcriptional activation of vir region, conjugation, DNA metabolism, intracellular transport, nuclear import, and tDNA integration. Injured plants secrete low pH sap which contains different compounds in acetylstringent, and it is the most effective one which is produced. These compounds serve as chemical attractants to the Agrobacterium and also stimulate vir gene expression. Similarly, sugars like glucose and galactose also stimulate vir gene expression. Now let's talk about the mechanism of infection. Acetylstringent binds with vir-A protein and activates it. Now vir-A activates vir-G gene which has DNA binding function. vir-G in turn activates other vir genes by phosphorylation. Next candidate is vir-D1 which has topoisomerase and endonuclease activity. It sits on right border sequence of tDNA. vir-D1 activates vir-D2 region which nicks right border region and remains bound to the 5' end generated. The 3' end produced at the nick serves as a primer for DNA synthesis in 5' to 3' direction. As a result, a single-stranded tDNA is produced with vir-D2 at 5' end of it. Now, vir-E2 is bound all over the single-stranded DNA, which protects the DNA from exonuclease activity. vir-V and vir-D4 proteins helps in the formation of a conjugal tube that mediates the transfer of tDNA along with VD2 complex into the plant cell. VD2 acts as a nuclear localization signal that drives the tDNA molecule into the nucleus of the plant. So this is the diagram, you can observe it nicely. So next step is integration. In the plant, single-stranded DNA is immediately converted into double-stranded DNA and integrates at random sites of the plant genome. After integration, genes of auxins, cytokinins and opines express themselves in, which results in uncontrollable growth in the form of tumor. There is a diagrammatic representation of whole process of infection, you can observe. Now scientists have realized very quickly that the TI plasmid has the capacity to transfer new gene into plant. That started the beginning of a research in development of TI plasmid as a vector. However, they found that DNA manipulation is very difficult, especially introduction of multiple cloning sites into the TI plasmid was found to be very difficult. So they developed two strategies to insert new DNA into the plasmid, and they are the binary vector strategy and co-integration strategy. Binary vectors are actually two plasmid systems. One contains vir and vir region and host specificity region, and this is the biggest plasmid. The smaller plasmid contains the tDNA and multiple cloning sites. If both the plasmids are present, it is good enough for plant transformation. So next strategy is co-integration strategy. So in this type of strategy, all types of genes are present in a single plasmid, and that is known as conjugation plasmid. This co-integration vector is produced by recombination among two intermediate vectors. One contains E. coli plasmid, variation, tDNA borders, and origin of replication, and PBR 322 sequences. The second vector contains gene of interest, some markers, and PBR 322 sequence. So this is a diagram. Since both vectors contain some part of PBR 322, homologous recombination will take place and a single vector will be produced within agrobacterium. This is the co-integration vector. Now next question is how to transform plant with TI plasmid. There are two options. First one is to infect the wounded plant with recombinant agrobacterium containing gene of interest such as drought resistance, pest resistance, etc., and let the cancerous growth to take place. Second option is to use plant biotechnology, in which first make plant cell suspension and co-culture with recombinant agrobacterium. As you know, recombinant vector contains certain selectable markers. You can select plant cells which have tDNA and gene of interest inside it. For example, if you use luciferase as a selectable marker, recombinant cells will glow in dark, but others will not. Now you grow them separately, and if you maintain proper in vitro plant tissue culture conditions, they will grow into plants with desired traits. So these are the references for further study. Thank you.